Imagine spending an entire day inside a closed room. The air feels normal, the temperature is comfortable, and you have everything you think you need: food, water, maybe a screen in front of you. Nothing feels immediately wrong. There's no alarm in your body telling you something is missing. And yet, quietly and gradually, something begins to shift. Not in your thoughts at first, not even in your mood, but deeper, in the behavior of your cells. The idea we're exploring is subtle, but important. Staying indoors for long periods may be depriving your cells of the signals they evolved to depend on. Not just the obvious things like oxygen or nutrients, but a much richer set of inputs: light, air movement, temperature variation, environmental complexity. These are not luxuries. They are part of the conditions under which human biology was shaped. We usually imagine cells as tiny machines, like little factories working inside us. Give them fuel, give them instructions in the form of DNA, and they carry out their tasks. But this picture is incomplete. A cell is not just a machine that runs on fuel. It is something that constantly measures, responds, and adapts. It is sensitive, it is reactive, it is always listening. A better way to think about a cell is as a kind of receiver. Like a radio that doesn't just play music, but constantly tunes itself based on the signals it detects. Light, temperature, chemical gradients, mechanical forces, these are all forms of information. Your cells are immersed in this stream of information, and they use it to decide what to do next. When to repair, when to divide, when to conserve energy, when to activate defenses. Now imagine taking that radio and placing it in a room where most of the signals are gone. The device still functions. It still has power. But the richness of what it can receive is reduced. It becomes limited, not because it is broken, but because its environment is simplified. Something similar may be happening when we spend most of our time indoors. The modern indoor environment is designed for comfort and stability. The light is controlled, the air is filtered and still, the temperature barely changes. From a human perspective, this feels ideal. But from a biological perspective, it is very different from the world in which our cells evolved. For millions of years, life existed in environments that were constantly changing. Light shifted throughout the day, air moved, temperatures rose and fell, surfaces were uneven. These variations were not disturbances, they were signals. They carried information about time, place, and conditions. And living systems, including human cells, evolved to depend on that information. When those signals are reduced, nothing dramatic happens immediately. You don't suddenly feel like something is wrong. That's what makes this idea difficult to grasp. The effect is not sharp or obvious. It's gradual, like a system that continues to run, but with less input, less variation, less guidance. So when we say that staying indoors may be starving your cells, we're not talking about starvation in the usual sense. We're not talking about a lack of calories or oxygen. We're talking about a reduction in informational richness, a quieter environment where many of the signals your body expects are no longer present. When we talk about staying indoors, the most obvious thing we lose is sunlight, but sunlight is often misunderstood. We think of it as simple brightness, something that helps us see. In reality, sunlight is a complex stream of information. It contains different wavelengths: ultraviolet, visible, and infrared, and each of these interacts with your body in a different way. Your body does not just ooze light, it responds to it. There are systems inside you that are constantly measuring light intensity, color, and timing. These systems help regulate what are known as circadian rhythms, internal clocks that control when you feel awake, when you feel tired, when hormones are released, and when cells repair themselves. You can think of these rhythms as a kind of timing system that keeps your biology synchronized with the world. Morning light acts like a reset signal. It tells your body this is the beginning of the day. As the day progresses, changes in light guide different processes: energy use, alertness, metabolism. And when darkness arrives, another set of processes begins: rest, recovery, cellular repair. Now imagine what happens when this signal becomes unclear. Indoors, light is often constant. It doesn't change much throughout the day. It lacks the full spectrum of sunlight, especially the ultraviolet and infrared components. Even when it seems bright, it is usually much dimmer than natural daylight. Your body is still receiving light, but the information it carries is incomplete. It's like trying to run a complex schedule with a clock that only shows part of the time. You can still function, but your timing begins to drift. Maybe not all at once, but gradually. Small mismatches begin to appear when you feel sleepy, when you feel alert, when your body expects food, when it expects rest. At the cellular level, this matters because many processes are time-dependent. Certain genes are activated at specific times of day. Hormones like melatonin and cortisol follow patterns that are closely tied to light exposure. Even immune function and cell repair are linked to these cycles. When natural light patterns are disrupted, these processes can become slightly misaligned. Not enough to cause immediate failure, but enough to create a kind of internal inconsistency. It's as if different parts of your body are no longer perfectly in sync with each other. There's also something interesting about the intensity of natural light. Even on a cloudy day, outdoor light is often far brighter than indoor lighting. Your eyes and brain are adapted to these higher levels. When you spend most of your time in dimmer indoor environments, your system receives a weaker signal about what daytime really is. This can blur the distinction between day and night in subtle ways. If your body cannot clearly distinguish between these phases, the transitions between activity and rest become less defined. And since many biological processes depend on these transitions, the effects can ripple through the system. What makes this difficult to fully understand is that light is doing many things at once. It is not just setting a clock. It is interacting with the skin, influencing chemical reactions, and affecting the brain directly through the eyes. Each of these pathways is complex on its own, and together they form a network that we are still trying to map. So when you stay indoors for long periods, you are not simply missing sunlight in a basic sense. You are reducing the complexity and clarity of one of the most important signals your body uses to organize itself in time. And the question becomes not just how much light you are getting, but what kind of light at what time and with what variation. Because for your cells, light is not just illumination, it is instruction. If light is one of the main signals your body depends on, then air is another, quieter, less obvious, but just as important. We tend to think of air in a very simple way. You breathe it in, you get oxygen, and that's enough. But the air around you is not just a supply of oxygen. It is a constantly changing mixture of gases, particles, moisture, and even microscopic life. And your body is sensitive to these variations in ways we are only beginning to understand. Step outside for a moment and notice what happens. The air feels different. Sometimes it's dry, sometimes humid. Sometimes it carries the scent of plants, soil, or rain. There's movement, wind that changes direction and speed. Even if you can't consciously detect all of it, your body is constantly registering these changes. Your lungs, your skin, and even your nervous system are responding to this dynamic environment. Now compare that to indoor air. Indoors, air is often still. It is recirculated, filtered, and controlled. This makes it comfortable and predictable, but also less varied. Carbon dioxide levels can slowly rise, especially in closed spaces. The range of natural compounds released by plants and soil is reduced. The subtle fluctuations that occur outdoors are largely absent. From a biological perspective, this matters because your body evolved in an environment where air was never static. It was always changing, always carrying new information. Cells in your respiratory system respond not only to how much oxygen is present, but also to how conditions change over time. Small shifts in oxygen levels, humidity, and airborne chemicals can influence inflammation, blood flow, and even brain activity. Think of it like listening to music. Outdoor air is like a complex, constantly shifting composition. There are variations in tone, rhythm, and intensity. Indoor air, by comparison, is more like a steady, unchanging note. It's not harmful in the immediate sense, but it lacks richness. And over time, that lack of variation may reduce the amount of information your body receives. There is also the question of microscopic exposure. Outdoors, you are constantly encountering tiny particles, microbes from soil, plants, and the wider environment. Your immune system interacts with these particles. Learning, adapting, and maintaining balance indoors, especially in highly controlled environments, this exposure is often reduced or altered. The system becomes less challenged, less engaged. This doesn't mean indoor air is bad in a simple way. In many cases, it protects us from harmful pollutants or extreme conditions. But it is different. It is simplified. And that simplification may come with trade-offs we don't fully understand yet. Another subtle factor is airflow. Moving air changes how heat is transferred from your body, how moisture evaporates from your skin, and how your respiratory system functions. These physical effects send signals to your body about the environment you're in. Indoors, where air movement is minimal or mechanically controlled, these signals are weaker or more uniform. The difficulty in studying all of this is that air is not a single variable. It is a complex system with many interacting components. When we try to isolate one factor, oxygen levels, for example, we miss the broader picture. In reality, your body is responding to the entire pattern, not just one element. So when you spend most of your time indoors, you are not simply breathing different air. In a basic sense, you are experiencing a reduced range of variation, a quieter stream of environmental information. Your cells still function, your lungs still exchange gases, but the richness of the input has changed. And just like with light, the question is not whether you can survive in that environment. Clearly, you can. The deeper question is how this quieter, more uniform atmosphere shapes your biology over time in ways that are subtle, cumulative, and not yet fully understood. There is another signal your body depends on that is easy to overlook because it feels so ordinary: physical interaction with the environment. Not exercise in the usual sense of running or lifting weights, but the constant low-level adjustments your body makes as it moves through the world. These adjustments are a form of information. They tell your cells where you are, how you are moving, and what kind of environment you are in. Step outside and walk on uneven ground. Even something as simple as a dirt path or grass introduces small variations. Your feet adjust, your ankles shift, your muscles coordinate in subtle ways. Your body is constantly recalibrating. Now compare that to walking across a flat indoor floor. The surface is predictable. Your movements become repetitive, almost automatic. The number of adjustments your body has to make is reduced. These differences may seem trivial, but at the cellular level, they matter. Cells are sensitive not only to chemical signals and light, but also to mechanical forces. When you move, stretch, compress, or shift your weight, those forces travel through your tissues. Cells detect these forces and respond to them. This process is known as mechanotransduction, the conversion of physical force into biological signals. For example, bone cells respond to stress. When bones experience varied loads, they adapt by maintaining or increasing their strength. Without that variation, the signal to maintain density becomes weaker. Muscle cells behave in a similar way, responding to tension and movement. Even cells in your blood vessels and connective tissue respond to the mechanical patterns of daily life. Outdoors, these forces are naturally varied. You step up, down, sideways. You adjust to slopes, textures, and obstacles. Temperature changes also play a role. A cool breeze or warm sunlight alters how your body regulates heat, how blood flows near the skin, and how your system balances energy. These are not extreme events. They are continuous, subtle variations that provide ongoing feedback. Indoors, much of this variation is removed. Floors are flat, furniture is standardized, and temperature is kept within a narrow range. Your body still moves, but in a more repetitive and constrained way. The signals that come from physical interaction become more uniform, less informative. Think of it like a conversation. Outdoors, the conversation is dynamic, full of changes and responses. Indoors, it becomes more like a repeated phrase. It still communicates something, but with less detail and less nuance. Your body is still receiving input, but the diversity of that input is reduced. There is also the question of posture and orientation. Indoors, many activities involve sitting for long periods, often in similar positions. This creates consistent patterns of pressure and tension in the body. Cells adapt to these patterns as well. Over time, the lack of variation may influence how tissues maintain themselves, how fluids move, and how systems coordinate. The challenge in understanding all of this is that these mechanical signals are subtle and continuous. They don't produce immediate, dramatic effects that are easy to measure. Instead, they accumulate over time, shaping how the body maintains its structure and function. We tend to think of movement in terms of intensity, how fast, how strong, how long. But there is another dimension: variability. The small, constant changes that occur naturally in a complex environment. These changes provide information that your body uses to stay adaptable. So when you spend most of your time indoors, you are not just reducing physical activity in an obvious sense. You are reducing the variability of physical signals your body receives. Your cells still respond, still adapt, but they are adapting to a narrower range of conditions. And once again, the question is not whether the body can handle this. It can. The deeper question is what happens when a system designed for constant variation is placed in an environment that is largely predictable, stable, and uniform. When we say that staying indoors may be starving your cells, it can sound dramatic, even misleading, because nothing about it resembles starvation in the usual sense. You can eat regularly, drink enough water, and still feel physically fine. Your body continues to function. There is no immediate crisis. So what exactly is being starved? To understand this, it helps to separate two ideas that we usually combine: energy and information. Food provides energy, calories that your cells use to carry out chemical reactions. But alongside energy, your body also depends on information. Signals that tell it when to use that energy, how to use it, and what priorities to set. Imagine giving a group of workers an unlimited supply of fuel and materials, but no instructions about what to build or when to build it. Work would still happen, but it might be inefficient, poorly timed, or misdirected. In a similar way, your cells can have enough energy, but still lack the guidance they need to operate optimally. This guidance comes from the environment. Light tells your body what time it is. Air carries subtle chemical cues. Movement generates mechanical signals. Temperature fluctuations provide context about conditions outside. Together, these inputs form a kind of continuous stream of information that helps your cells organize their behavior. When you spend most of your time indoors, many of these signals become reduced or simplified, not eliminated, but less varied, less dynamic. The result is not a shutdown, but a narrowing. Your cells continue to function, but with a smaller set of inputs to guide them. A useful way to think about this is through the idea of sensory richness. Consider the difference between listening to a full orchestra and hearing a single repeated note. Both are sound. Both can be detected. But one contains far more information than the other. It has layers, variation, timing, contrast. The other is simple and unchanging. Your body, in a natural environment, is exposed to something like that orchestra, a complex mix of signals that change throughout the day. Indoors, the experience is closer to the repeated note. It is stable, predictable, and limited in variation. This doesn't mean that indoor environments are harmful in a direct or immediate way. In many cases, they protect us from extreme weather, pollutants, and physical danger, but they also remove a layer of complexity that your biology evolved with. The idea of starvation here is about missing variety, missing context. It is a kind of undernourishment of signals. Your cells are still receiving input, but the range of that input is smaller. And over time, systems that rely on variation may begin to adapt to this simpler environment. Adaptation is an important part of this story. The body is not passive. It adjusts to the conditions it experiences. If the environment becomes more stable and predictable, the body may become more specialized for that stability. But specialization often comes with a trade-off: reduced flexibility. In a highly variable environment, flexibility is valuable. Your system learns to respond to change. In a stable environment, that flexibility may not be used as much. The system becomes efficient within a narrow range, but less prepared for variation. This is why the effects of staying indoors are not always obvious. The body doesn't fail, it adapts. It finds a way to function under the available conditions, but that adaptation may come at the cost of reduced responsiveness, reduced synchronization, or reduced resilience. What makes this difficult to grasp is that we are not used to thinking about health in terms of information. We think in terms of nutrients, exercise, sleep. These are important, but they are only part of the picture. There is also the question of what signals your body is receiving from the world around it. So when we talk about cells being starved, we are pointing to something less visible, a gradual reduction in the richness of the signals that guide biological processes. Not a lack of fuel, but a lack of complexity. And the deeper question that follows is this: how does a system built for a rich, changing environment behave when that richness is quietly removed? When we try to understand all of this scientifically, we run into a problem that is not obvious at first. It's not that we have no data. We actually have a lot. We know that light affects hormones. We know that air quality influences cognition and inflammation. We know that movement changes how tissues grow and repair. Each of these areas has been studied in detail. And yet, when we try to put everything together into a single, clear explanation, it becomes difficult. The reason is complexity. Your body is not responding to one signal at a time. It is responding to many signals simultaneously, and those signals interact. A change in light exposure can alter your sleep. Changes in sleep can influence hormone levels. Hormones affect immune function, metabolism, and even mood. At the same time, air quality and physical movement are also influencing these systems in parallel. Everything is connected, and the connections are not simple or linear. This creates a challenge for how science is usually done. In order to study something, we tend to isolate it. We change one variable while keeping everything else constant. This works well for simple systems, but it becomes limiting when the system depends on interactions between many variables. If you study light in isolation, you learn something important. If you study air or movement separately, you learn something else. But the real behavior of the body emerges from how all these factors combine. There is also the issue of timescale. Many of the effects we are talking about do not happen instantly. They develop slowly over days, weeks, or even longer. Small changes accumulate. A slight disruption in circadian rhythm here, a small reduction in environmental variability there. Each change might be too subtle to notice on its own, but together they form a pattern. This makes it difficult to design experiments. In a laboratory, it is easier to measure immediate, clear effects. It is much harder to capture slow, cumulative changes that depend on a complex, real-world environment. As a result, some of the most important questions remain only partially answered. Another limitation comes from our own perspective. Humans understand the world through language categories and simplified models. We like clear explanations, cause and effect. This leads to that. But biological systems don't always behave that way. They are adaptive, nonlinear, and context-dependent. The same signal can produce different effects depending on timing, intensity, and the presence of other signals. This is why the idea of staying indoors affects your cells can feel vague or unsatisfying. It does not point to a single mechanism or a single outcome. Instead, it suggests a shift in the overall pattern of inputs your body receives. And patterns are harder to describe than individual variables. There is also a deeper issue. The environments we evolved in are not easy to recreate under controlled conditions. Natural settings are full of variation, changing light, moving air, diverse surfaces, complex ecosystems. Indoors, we simplify these variables to make them manageable. But in doing so, we may remove the very interactions that matter most. So we end up in an unusual situation. We have strong evidence that different aspects of the environment influence biology. We have clear mechanisms for some of these effects, but we lack a complete, unified framework that explains how all these pieces fit together in everyday life. This is not a failure of science. It is a reflection of the kind of system we are dealing with. Living organisms are not simple machines. They are dynamic processes shaped by continuous interaction with their surroundings. To fully understand them, we may need new ways of thinking, approaches that focus not just on individual factors, but on relationships and patterns. In the meantime, we are left with an incomplete but meaningful picture. We can see that environment matters, that signals matter, that variation matters. What we are still learning is how these elements combine to shape the behavior of cells over time. And perhaps the most honest answer is this: we understand enough to know that something important is happening, but not enough to describe it completely. If we step back and look at the bigger picture, staying indoors forces us to reconsider what it means to be alive and how we are connected to the world around us. We often think of ourselves as separate from our environment. There is the onside, where we exist, and the outside, which is the world we observe and interact with. But at the level of cells, that distinction begins to blur. Light enters our eyes and skin, air enters our lungs, and signals continuously flow between us and our surroundings. We are not enclosed systems. We are dynamic processes embedded in a web of environmental inputs. When we spend long periods indoors, we are not merely changing our location. We are changing the quality and diversity of signals that shape our internal processes. The richness of natural light is replaced with artificial light that lacks variation. The constantly shifting air outdoors is replaced by filtered, often stagnant indoor air. The complex mechanical challenges of outdoor movement are replaced with flat, predictable floors and repetitive motions. Each of these changes narrows the stream of information our cells use to organize themselves. Over time, this reduction in informational richness may subtly alter how our bodies function. It does not cause immediate failure, but it may reduce flexibility, responsiveness, and resilience. Systems designed to handle variability and change may begin to adapt to the simpler, more uniform indoor environment. Adaptation is a hallmark of life. Cells adjust to the inputs they receive, but adaptation to reduced complexity comes at a cost. The system becomes less prepared for the diversity of conditions it was originally designed to navigate. This is why the concept of starving your cells is useful, even if it is not starvation in the conventional sense. It is a form of undernourishment, an underexposure to the environmental signals that guide biological processes. Nutrients, oxygen, and calories may be plentiful, but the context in which cells operate is constrained. Information and variability are essential forms of nourishment that are easy to overlook, yet they shape everything from hormone rhythms to immune responses to cellular repair. Understanding this changes how we see ourselves in relation to the environment. We are not merely contained within our skin. Life is not a self-contained process. Our bodies are tuned to interact with patterns of light, air, movement, and variation that extend beyond ourselves. Our cells, in their quiet and relentless way, are listening and responding to these patterns continuously. By simplifying our environment, we simplify the feedback that keeps our biology dynamic and robust. This perspective also reframes health. It is no longer solely about nutrition, exercise, or rest. Health is about the quality of the inputs we receive from the world, the environmental signals that allow our systems to function optimally. Light, air, mechanical variation, and environmental richness are not peripheral. They are integral to the maintenance of life. They are part of the language in which our cells understand the world. The philosophical implication is profound. If our cells are continuously shaped by the environment, then reality is not only external but participatory. Our bodies are not isolated objects but processes in ongoing conversation with the world. Every step outside, every breath of fresh air, every shift in sunlight reconnects us to a larger network of signals that we evolved to depend on. Staying indoors reduces that conversation. Being outdoors restores it. Ultimately, this challenges the way we perceive boundaries between ourselves and the world. We are not separate observers. We are integrated participants in an environment that provides the signals necessary for our bodies to thrive. Life is not only in the cells, it is in the ongoing interaction between cells and environment, an intricate dialogue that reveals how deeply connected we are to the patterns of reality itself. By seeing our bodies as processes rather than static machines, and the environment as a source of essential signals rather than a mere backdrop, we gain a new perspective on what it means to be alive. Staying indoors may be comfortable, but it gradually simplifies the conversation between you and the world. Stepping outside is not just a choice. It is a reconnection with the very signals that allow your cells to live fully and your body to experience the richness it was designed to perceive.